Started. So we're going to start a new section today. Oh, let me remind you first. We have an exam in here a week from today that will cover digestion, which is the section we're going to start today. We'll cover respiration, cover the renal system. And we'll try to get those practice exams mounted here in the next day or so. So where I want to begin, though, is I want to finish off the story on the renal system. And I'd gotten this far, I'd gotten through this description in the first lecture, but I didn't get to it in here. So I want to just remind you here quickly that the vasa recta is a capillary. As such, it's a capillary that originates as a branch of the efferent arteriole. So the efferent arteriole is coming out of the glomerulus, and then it branches. And in some cases, it branches into a peritubular capillary around the proximal convoluted tubule. Some cases, it branches into a peritubular capillary around the distal convoluted tubule. And in other cases, it will form a branch that now becomes the vasa recta. In all cases, once it branches, it now becomes a proper capillary. And here, this example is the capillary of the vasa recta that runs in parallel with the juxtamedullary nephron. So all along the juxtamedullary nephron, we see right next door this vasa recta. Now, the way I presented it in the discussion to the first section was that the fluid running through the vasa recta is running in the opposite direction of the fluid running through the tubule. And so that ends up generating a countercurrent system. In biology, you don't really need to do that, but I think if you see the fluid running through the capillary going in the opposite direction of the fluid running through the nephron, it's much easier to see that countercurrent arrangement and recognize how the vasa recta is maintaining the gradient, the corticomedullary gradient. The corticomedullary gradient is essential. If you put your phone away, I'd appreciate it. The corticomedullary gradient is essential for concentrating urine and doing the final stages of increasing the urea concentration deep in the medulla. All that is done. It's generated by the juxtamedullary nephron. But now the player here for maintaining that corticomedullary gradient is the vasa recta. If you block blood flow through the vasa recta, this corticomedullary gradient breaks down in a matter of hours, maybe minutes. And once you've broken down the corticomedullary gradient, you can no longer use that gradient to make concentrated urine. So you end up with basically isosmotic urine. And so you need to have this structure to maintain the gradient. But that's all the more I'm going to say about that. Hold on to the idea that it's important for maintaining the gradient. Hold on to the idea that it's capillary. Hold on to the idea that it's blood flow through it and that it's highly permeable. What we want to spend most of our time on today is the beginning of a discussion of the gastrointestinal tract. And we'll begin this story with something that seems pretty obvious to all of us in asking the question of what is food? And when you actually break down what food is, it's pretty simple. There are only a handful of components. Even though we may think of food as being this complex mixture of all kinds of things we eat, when you break it down to its fundamentals, there are only a few components. The first component is carbohydrate. And we're going to see the carbohydrate can come in several forms. But the carbohydrate is almost entirely, one way or another, made from 6-carbon sugars called hexosugars. We'll begin discussion here with something known as polysaccharides. The polysaccharides are polymers of glucose, and glucose is a hexosugar. It's a hexosugar, it is composed of 6 carbons. So we're going to generate these polysaccharides from the 6-carbon glucoses, and we're going to see that the polysaccharides that we have mostly in our diet come as starch. But starch is composed of something called amylose and amylopectin. Now, in the end, all we are looking at here is that starch, whether we're thinking about it as amylose or amylopectin, is a polymer of glucose. As we walk our way through this, we'll look at some of the chemistry, and we'll see that amylose is a straight chain series or linkage of glucoses, all linked by something known as 1,4 glycosidic bonds. We'll take the number 1 carbon of one glucose, link it to the number 4 carbon of the neighboring glucose, and we'll get these straight chains of amylose. But one of the things that we find in starch is that in addition to these straight chains, there are branched chains. The branched chains are called amylopectin. Again, it's using the number 1 carbon of each of the glucoses. But now we're going to use the number 1 carbon of one glucose and the number 6 carbon of a neighboring glucose, and now we'll form these so-called 1,6 linkages. And that ends up making not only these great long straight chains of the amylose, but a highly branched structure of amylopectin. And we'll walk our way through that. So these may be made up of literally millions of glucose molecules. But another major component of the carbohydrate coming into our diet are these so-called disaccharides. And the disaccharides come as 3 major forms. There's maltose, which is glucose plus glucose. Again, as the name implies, a disaccharide has 2 sugars. And in each of these cases, they are 6 carbon sugars. So maltose is the 6 carbon sugar of glucose linked by a 1,4 glycosidic bond to another glucose. So that's glucose plus glucose is maltose. Sucrose, another molecule that we like a lot because it tastes really sweet, is glucose plus fructose. And then finally, there's this molecule lactose. Lactose is unique to mammals. Mammals, after all, suckle their young with milk, and one of the major sugars in milk is lactose. Lactose is a disaccharide. It's composed of a molecule of glucose plus a molecule of galactose. In almost all mammals, after the time of weaning, this molecule isn't any longer digestible. But we'll see in humans, some of us have a mutation that allowed this to happen, and we'll talk about where that came from. And then finally, there are the monosaccharides, and the monosaccharides are the hexose sugars, and the 3 major hexose sugars are glucose, fructose, and galactose, all 3 of which are incorporated into the disaccharides to form maltose, or sucrose, or lactose. Then another major component of the diet that we're all familiar with, of course, is proteins, but proteins are simply polymers of amino acids. And as we work our way through this, one of the things you'll begin to recognize is that in our diet, we take in these gigantic molecules, these great big polymeric molecules, and as part of digestion, we'll take these polymeric molecules and convert them into their component parts and absorb the component parts across the epithelium in a fancy form of epithelial transport. But we have spent a whole lot of time in here talking about proteins, but our emphasis so far has been in forming peptide bonds so that we could generate the polymers of amino acids to make these gigantic molecules called proteins. And we talked about how we could select each amino acid individually using the mechanisms of translation. We're just going to break these things down pretty indiscriminately as we digest them. But in the end, we're going to be doing everything in reverse. We're going to be breaking the peptide bonds to generate individual amino acids, because it's the individual amino acids, for the most part, that are going to be absorbed across the epithelium of the small intestine. Another major player of the food we eat, and of course we all know this is fats or lipids. Most of the fats and lipids we take in come in as triglycerides. And you'll remember from our early discussions that a triglyceride is made of a single molecule of glycerol, that 3-carbon alcohol, linked by ester bonds to fatty acids. The fatty acids are normally 16, 18, 20, or 22 carbons long. And the reason that they come in these even numbers is because of the biochemistry of the way we generate these fatty acids. These fatty acids are generated in this way because we use 2 carbon chains called acetate. And these acetates then, from acetyl-CoA, are then linked one to the next through the carbon-carbon bonds to make these multiple carbon chain lengths, but they're all even because they come 2 carbons at a time. Now, if you get into the story of cattle, and some of you will do that here because you're in animal science, you'll find it doesn't come out so neatly because of the peculiar characteristics of cattle lipid digestion and the way they'll actually generate methane and then incorporate the methane into fatty acids. You'll actually get odd chain lengths, but let's not deal with that. Let's think mostly just of these even numbered chain lengths. Nucleic acids, important part of a diet. We're going to skip right over it. We take in RNA and DNA. Anything we eat We eat living things, whether they're plants or animals, and they all contain DNA and RNA. So this is also a component of the diet, and we'll break those down into their component parts as well. And then finally, where I want to finish is just an introduction to the three major concepts to digestion. When you think about digestion, and it's worthwhile wherever we're talking about along the GI tract to devolve the digestion down to these three major ideas. First is motility. Whether it's chewing in the mouth or whether it's moving the stomach contents back and forth as a kind of churn, it's motility. Or whether it's moving the bolus of food you've eaten through the small intestine, that's motility. And so that's an important characteristic of digestion. Secretion also becomes an important characteristic, and we'll talk a little bit about that today when we talk about saliva. But all along the GI tract, you're moving molecules across cells. And one of the major things you're going to be moving are digestive enzymes and hormones and all manner of things. So there is a tremendous amount of secretion along the GI tract. And finally, what we're trying to do in the end is absorption. So in the end, what we're trying to do is generate these small molecules from the big chains, generate individual molecules and absorb those across the epithelium. But if you hold on to those three major ideas, it will serve you well as we talk about the different regions of the digestive tract. But let's start here with simply the idea of what is food. And we'll begin with the concept of the three major characters of carbohydrates. The term carbohydrate dates all the way back to the 18th century, and in the early days when they first beginning to understand you could burn things and that oxygen was required, they were burning anything they could find. And when they would burn these molecules, particularly like starch or even cellulose, they would find that the byproduct was water and CO2. Therefore, the term carbohydrate. That's where it all comes from. We now know that these carbohydrates are made mostly as polymers of 6-carbon sugars. The most common of the 6-carbon sugars we're going to see is glucose. If we look at one of these carbohydrates, we can see that one of the most common is starch. And that starch comes in two varieties, as amylose and a related molecule called amylopectin. But in addition to these polymers of carbohydrate, such as starch, which we call a polysaccharide, poly simply meaning many. And when we talk about many here, we mean lots, because these starch molecules can be polymers of several million molecules of glucose. They're enormous molecules. But in a related nomenclature, even though this may be a really enormous number, we also have disaccharides. Di meaning two. The disaccharides are composed of two hexose sugars. These can come, as we said on the sideboards, as maltose. That's glucose plus glucose. Or as sucrose, which is glucose plus fructose. Or the third character here, lactose, which is glucose plus galactose. This, of course, is milk sugar. But let's look a little bit at these polymers of glucose. Let's try to draw a molecule of glucose here as best I can. 6 carbons, it's the first carbon, and it's on a 6-membered ring. We can number the carbons, 1, 2, 3, 4, 5. The number 5 carbon is linked to the number 1 carbon through an oxygen, and up here is the number 6 carbon. Each one of these carbons has a hydroxyl and a hydrogen, hydroxyl, hydrogen, OH, OH, and an H. And up here, there are 2 hydrogens at the number 6 carbon and an OH. And also 1 hydrogen out here. So from this, with all of these hydrogens and hydroxyls and the carbons, you can pretty much see how when we burn these molecules, we end up with carbon dioxide and water. When we add oxygen through burning, we make carbon dioxide and water, and that's the term carbohydrate. Now, the beauty of this molecule is that when we look at this ring structure, 1, 2, 3, 4, 5, we can now take this number 1 carbon and line up over here a number 4 carbon. This is always hard for me. So 1, 2, 3, 4, 5, and 6. Now, what I'll emphasize here is there's this hydrogen out here on this carbon. Now, what we can do is take this hydrogen and this hydroxyl, and we generate water. But in the end, what I want you to recognize is we now have this carbon-oxygen-carbon bond. This is called a glycosidic bond. And we now can generate this over and over and over again. We can out here again take this number 1 carbon, link it through an oxygen to a carbon. And again, 1, 2, 3, 4, 5. And now have this 1, 4 glycosidic bond. We can do it over and over again. If we start here at this end and keep adding glucoses, we can make this hundreds of thousands of glucoses long. And we can continue this out in a straight chain. If this is a straight chain of glucoses, straight chain of glucose is linked by 1, 4 linkages, we're going to see that this is in fact a molecule of amylose. Let me put a caveat in here, linked by alpha 1,4, and we'll kind of come back to this alpha story in a minute. So they're linked by these 1,4 linkages. You make a great long straight chain, and that now is amylose. But we can also take this number 6 carbon up here, which has the hydrogen, as a hydroxyl, and 2 hydrogens. We can now take this, let me do it this way, H, OH, and hydrogen. Now we can utilize this hydroxyl and also now take this hydroxyl to a number 1 carbon. 1, 2, 3, 4, it's carbon. 5, there's the oxygen, there's the number 6 carbon. Now what I want to emphasize here is that we can now take this number 6 carbon and link it to the number 1 carbon. So now we have another glycosidic bond. This is said to be a 1,6 glycosidic bond. These glycosidic bonds, now we can make this, now we can take another glucose here and make a 1,4 linkage, so we can make this great long chain out here. When we start to make these chains utilizing the 1,6 linkages, we now have something called amylopectin. So amylose is straight chain, 1 to 4, 1 to 4, but when we start adding in this branching off the number 6 carbon, then now we formed amylopectin. But that's a detail of the chemistry. What I want you to recognize is that we have these great polymers of glucose molecules. And this glucose, as we work our way through this, is going to be one of the major carbon sources that we're going to feed into the mitochondria to generate ATP. And this is a great storage molecule for that. And potatoes do a lot of storage as starch. Well, where did it go? There. Okay. What we're seeing here is just a pretty picture of what I put on the board. This is the 6-carbon structure of glucose. And if you start getting into this in organic chemistry, you're going to see there's a whole bunch of there are several stories in here about the chair formations of the 3-dimensional shapes. I don't get into that. What I do want to show, though, is we have this 6-carbon structure. Each of the carbons has a hydrogen and a hydroxyl. And out here, we have a number 6 carbon sort of dangling out in air. And then 1 carbon is linked to the 5 carbon through the oxygen. That leaves open this number 1 carbon. And what this allows us to do, as we showed on the board, is it allows us to link the number 1 carbon of one glucose molecule to the number 4 carbon of a neighboring glucose molecule. And we can link them through these oxygens. And we can do this over and over again, making this great long straight chain to get amylose. Now, there's an added caveat I want to put in here, in that the organization of this oxygen bond is such that these oxygens always reside below the plane of the ring of the 6-membered ring of this 6-carbon structure. So these are always below the ring. That makes amylose. Now, that seems like a trivial difference. But if we go to this molecule is cellulose. And you'll see it pretty much is the same thing. Here we have a 6-membered ring. It's linked the number 1 carbon to the number 4 carbon of the neighboring glucose molecule by this oxygen. But now we find that rather than all of the oxygens being below the plane of the ring, these alternate, below and above, below and above. That changes the 3-dimensional shape of the molecule in such a way that, for reasons I don't understand, most organisms have never generated enzymes to break this molecule down. Now, we can digest a little bit of cellulose, but as we do it, we're using bacteria in our large intestine. Animals that have developed means of breaking down the cellulose have highly specialized mechanisms, and it's always through a symbiotic organism. Either a bacterium that has the cellulase molecule that can break these bonds, or an individual eukaryotic cell called a protist that breaks them down. Now, if you're a cow, what you do is you have all of these protists and bacteria in your rumen, and they will then digest these cellulose molecules so that you can actually generate glucose from these great long chains of cellulose. Now, if you ever get a chance to do this, and this is something that's unique to a veterinary school, is that on this campus we maintain a cow called a fistulated cow. And the fistulated cow has in its flank a piece of plastic, basically a cylinder of plastic. And you can put on a glove. I suggest you put on a glove and put it up to your shoulder. And you can reach inside the rumen of this animal. And it's astonishing. There's a barrel inside the abdomen of this animal, and it's full of fluid. And that fluid is the rumen. And that specialized structure is called the rumen. And what's in that rumen is this complex mix of the digesta, what the cow has eaten and chewed as it's cud and swallowed and fed into the rumen, along with all of these individual single-celled organisms that are capable of breaking these bonds. This is true in all the ruminants. It's true whether you're a cow or a goat or a sheep or a deer or an elk or a reindeer. They're all ruminants, and they'll use the rumen to break these things down. Now, if you're a horse, you do it differently. You can break down, if you're a horse, you can get glucose out of cellulose. But horses are what's said to be post-gastric fermenters. And they have a region of the large intestine called the cecum. And the cecum is just packed full of these protists, which then are doing the job of breaking down the beta linkages in the cellulose molecules. Even if you're a termite and your livelihood depends on eating wood, you have to have in your gut protists that are doing the job of breaking down the cellulose. Otherwise, you can't break it down. And why biology, as eukaryotic organisms, has never figured out a way to break down these beta-1,4 linkages, I don't understand. But it seems to be something that only unicellular organisms can do. Now, this other figure here is simply to flesh out the idea of the difference between the straight chain and the branch chain of the amylose versus the amylopectin. Again, here are these 1,4 linkages. If we simply had this great long stretch of these 1,4 linkages, that would be amylose. But now once we take this number 6 carbon out here on this glucose and link it through a glycosidic bond to the number 1 carbon up here on this glucose, and then begin to make 1,4 linkages out in this direction, now we have amylopectin, and so this is going from the 1,4 bond to the 1,6 bond. If you put your phone away, I'd appreciate it. So with that, are there any questions? So as we go through this, I'll hold on to this and come back to it. What we're going to do is we break these chains down. We're going to break them down into maltose, which will be 2 glucose molecules linked by these 1,4 linkages. And that will be the initial breakdown of starches. But let's just take a minute and remind ourselves of what proteins are and how proteins are an important part of the diet. Proteins are polymers of amino acids linked by peptide bonds. We spent a lot of time describing how we form peptide bonds, how the ribosome is an enzyme, effectively, that forms those peptide bonds. We're going to run that process in reverse here. And when we digest proteins, we will digest proteins into amino acids. Another example of how we take in these polymers, these great big molecules, and we eat them as big molecules, but then in the process of digestion, we break them down into their component parts, and the component parts are amino acids for proteins and glucose for the polysaccharides. We've spent so much time on this, I think we can leave that behind, but let's spend a little bit of time on lipid. And in our diet, lipid is mostly triglycerides. Whether these are saturated fats that come from animals, which will be lard, or whether they're unsaturated fats, such as the vegetable oils, they're still triglycerides. And I'll remind you from the early days of our discussions in here that they're generated from a 3-carbon alcohol called glycerol. And remember that we employed this alcohol along with the carboxylic acid end of these great long structures called fatty acids. We said that the fatty acids are polymers polymers of acetate. And what we mean by acetate is simply the 2-carbon chain. And typically we find these fatty acids, because they're polymers of acetate, being 16 18, 20, or 22 carbons long. I'm not going to draw this all out, but you can see how that we have these two carbons and so forth, all in even numbers out to 16, 18, or 20, or 22. Now, I've shown this as a saturated fat, where each of these carbons has two hydrogens attached. But although this is true for most animal fats, vegetable oils are organized a little bit differently. Vegetable oils typically will have a double bond. Sometimes they'll be polyunsaturated. There'll be several of these carbon-carbon bonds which are double bonds rather than single bonds. But again, if you count the numbers, 1, 2, 3, 4. Each of these carbons has 4 bonds. Now, in most cases in biology, these hydrogens are on the same side of the chain. This is said to be the cis configuration. But in some cases, we'll find these hydrogens are on the opposite sides of these double bonds, and this is called the trans configuration. This trans configuration doesn't happen very much in biology, but as good human chemists, we can make these. And it was found that if you would make trans fats and put trans fats into processed food, that you could make processed food like cakes and sweets and such, put them on the shelf, and they would stay fresh for weeks or months at a time. And one of the reasons they would stay fresh for all that time is that other organisms were unable to deal with these trans structures of the hydrogens. Similarly, we can't deal with these trans fats either. And it's been recently found in the last few years that the statement by the dietitians is that there is no safe level of trans fats. And the reason is we think that because we can't digest properly, we can't break these molecules into their component parts, we end up depositing them in any number of places. And one of the places they're worried about is you begin to deposit them along your coronary arteries, and you end up with plaque. And so for that reason, over the last few years, we've tried very hard to eliminate these trans fats from our diet. In fact, the statement is there is no safe level of trans fats, but there is a safe level of radioactivity. I find that peculiar that we would add these to our foods. But you have these great long chains, and you can do this for each of the 3 chains. Remember, this is the ester bond. Now, one of the things we'll find as we move our way through this and talk about digestion and how we're going to convert these into their component parts, that proteins and carbohydrate are fairly easy to deal with. After all, they're water soluble. So we can use water-soluble enzymes and attack these gigantic molecules with water-soluble enzymes and break them into their component parts. And that's not true here. One of the things we're going to find is that when we start dealing with fats, because they're water insoluble, they have to be handled in a particular way. And we go to great lengths to try to digest these things. But it's really quite worth our while because the energy that we see compressed into these molecules is tremendous. We can clip off these 2 carbon units at a time, take those acetates, plug them into the mitochondria, and make ATP. So this is a very efficient storage mechanism for structures we can convert into ATP. We can also store these things away in our adipocytes as lipid. So they're a great storage system, and we pay a price for the difficulty in handling them. But because there's so many acetates in here, in each one of these molecules, it's worth our while. I mean, if you think about this for a moment, if you simply take 20 carbon chain length here, that's 10 acetates. 3 chains, that's 30 acetates. So you have 30 acetates for every triglyceride that you can now feed into the Krebs cycle, the citric acid cycle and generate ATP. So these are great storage molecules for high caloric molecules. We're not going to say anything about nucleic acids, but any questions up to here? The long and the short of this is breaking big molecules into small molecules. But let's begin to think about this a little bit, and I'm going to lay out the three major concepts of the GI tract. And wherever we are in the GI tract, whether we're in the mouth or whether we're in the small intestine or the stomach, it's worth thinking about these three major concepts because I think it sort of orders our thinking on what's going on at each of these levels. And we'll begin here with something we're all familiar with, basically eating food. And we'll start with this list of motility, secretion, and absorption. We can sort of head this as concepts of digestion. And the concepts are motility, secretion, and absorption. In fact, absorption is going to be almost entirely relegated to the intestinal epithelium. We get very little absorption of molecules across the stomach or across the mouth and esophagus. But nonetheless, I want to start here with something that we're all familiar with. Just the idea of going to eat lunch. And when I begin this, and I apologize for this, this is going to look a little bit like a nephron. I don't mean it to be a nephron. What I mean it to be is a mouth and a pharynx and an esophagus. And to differentiate it, we're going to have some teeth. But we know from everyday life that one of the first things we do with the mouth is motility. And that's chewing. And one of the things that chewing does is that it increases the surface area of the food. No surprise. Go out for lunch, you're going to take a bite, you're going to chew it up, and you're going to chew it until you increase the surface area. And then you're going to take your tongue and you're going to push that, what you've chewed, back up against the soft palate at the back of your mouth, and that's going to start a reflex, which then starts the muscle contraction. In the esophagus. So this idea of swallowing the idea of swallowing and chewing is just a classic form of motility. In chewing, we activate our masticatory muscles and we chew the food, then we swallow it. That's just motility. But also in the mouth, there is a form of secretion that we're all aware of, and this is saliva. And if you haven't had lunch yet, you may simply think about lunch, and by thinking about lunch, this will begin the salivation process. So I want to spend a moment here talking about secretion of saliva. Now, when we secrete saliva, it basically comes in two forms. It's called serous, and this is watery. And then there's a mucus form. And these contain mucopolysaccharides. This way. Now, if you get down into the weeds of this, you'll find that there are some of our salivary glands that are almost entirely dedicated to generating this watery or serous saliva. Others are dedicated to making the mucus. But after all, and we know this intuitively, if you take a bite of cracker, you know you'd better moisten it before you swallow it. And you also know that as part of the swallowing, you're going to make it slippery, and that's the mucus. But also, in addition to the secretion, particularly in saliva, there will be something called salivary amylase. ASE tells you it's an enzyme. Amylase is going to break down amylose. So what salivary amylase does, it converts amylose to maltose. So we'll take these great long chains of glucoses and break them down into two glucose chains called maltose. I think it's worth taking a few minutes here to describe how saliva is generated, not just in terms of the saliva part of it, but because we can bring around several ideas we talked about in other units, particularly the renal system, about how we can do secretion. So I want to spend a moment here on, do it over here, saliva secretion from salivary glands. These salivary glands are classic exocrine glands. And when we think about this, we can draw the glands that we have talked about, the exocrine glands we've talked about earlier in the course. And I'm going to put this in as a series. I'm going to try to make it as bulbous as I can. If you look at the histology of salivary glands, you see they're highly branched. But I'm going to simplify it so we have sort of this bulbous structure here, made of a continuous layer of epithelium. And this empties now out on the surface. So as we've seen before, this is a surface epithelium. This is a duct epithelium. This is, in fact, a secretory epithelium. But in this particular case, we call these acinar cells. Again, to orient you, as with all epithelia, these sit on a basal lamina. And if we look carefully at these acinar cells down here, we find they have characteristics very similar to what we saw in the proximal convoluted tubule. So down here is going to be basilar. So we can imagine that faces on this side a capillary. Out here is the lumen of the mouth. And the direction of flow of fluid is going to be here from the lumen of the tube up through the duct and out onto the surface. A classic exocrine gland. So on this side is the apical membrane. And this is said to be the lumen of the gland. as before, we see that these epithelial cells are stuck together by tight junctions. And then right down here, right below this cross, where these two cells come close together, into this narrow opening, we find on this membrane a sodium-potassium ATPase. The net effect here is to pump sodium out into this extracellular environment. Just as we saw before, now we're going to have an osmotic gradient. And water is going to move from the extracellular environment between the cells and come out here into the lumen of the gland. So water is going to be coming this way. Now another old friend that we have here is on these basilar surfaces of these membranes will be the sodium potassium 2-chloride transporter. So we're moving sodium and chloride and potassium into the cell. And then right in these regions, we're pumping the sodium out again to form an osmotic gradient so we can draw water across. But if you were to follow, and chloride will follow. So if you were to just look at this, though, you would say, well, we're going to get an isosmotic solution in here of sodium chloride. And you would predict from that simple model that saliva would be salty. Saliva isn't salty. We all know this. We know that saliva has little salt in it. So where does the salt go? Well, we use the salt to generate an osmotic gradient. But then these duct cells, the duct epithelium, play the role of removing sodium. So one of the characters along these cells will be the enax. Remember, we saw enax in the distal convoluted tubule. And so now we can help remove the sodium simply down the concentration gradient by letting the sodium enter the cell. Also, what we'll find is that there are sodium-potassium pumps, which are going to move sodium into the cell. And in the end, we end up having potassium out of the lumen. And the chloride gets removed here, but replaced with bicarbonate. So what happens by the time the saliva gets out into our mouth is we now have a dilute solution of potassium bicarbonate. And that all happens because of the characteristics of these epithelial cells. The glandular epithelium, the acinar cells, are moving sodium chloride through pumps and through these transporters, but that generates an osmotic gradient to draw water into the lumen of the gland. But then to recycle the sodium and the chloride, we use the duct epithelial cells to remove the sodium and chloride. But in the process of removing the sodium and chloride, we exchange the chloride for bicarbonate, and we exchange the sodium for potassium, and we end up with potassium bicarbonate. That's how we generate saliva. Now, this story would be a little bit different if we were talking about those glands making the mucus-rich saliva. In those cases, we'd actually be actively secreting mucopolysaccharides into the lumen to make this mucus-thick saliva. And that's now important, though, in making saliva, making the food slippery so that we can, in fact, swallow it. Where we're headed on this is we're now making the food wet and slippery, chewing into smaller pieces, and then bringing it back into the soft palate, starting the reflex for swallowing. And we're all familiar with this reflex for swallowing. We use it all the time. We know it's a reflex. And one of the ways we know that is that if you've ever been chewing gum, and chew gum for a while, and you make a mistake, and you get it back too far toward the back of your mouth into that soft palate, it's gone. There's nothing you can do because you've started the reflex to begin the muscle contraction to force that chewing gum down your esophagus and into your stomach. let's leave it there, and next time we'll talk about how the organization of muscle